There were several methods used to extract and purify the lower termite DNA and the diverse protists that are carried in their lower gut. We began the lab by doing a DNA extraction using lower termites from the Papago Park in Tempe, Arizona. We extracted the lower gut using forceps and placed the gut into the Riener solution to open it up. We stored the sample at negative 20 degrees Celsius for a day and a half, and then broke apart the protist to collect the DNA from the sample. Now we placed the sample into a tube and added 180 microliters of digestive solution and 20 microliters of proteinase K. We then incubated the sample at 56 degrees Celsius for half an hour with breaks every 15 minutes for it to be vortexed. After incubation is complete, we add 20 microliters of RNA solution to the sample and then incubate it again at room temp for 10 minutes. Next, we added 200 microliters of lysis solution to the sample and vortexed for 15 seconds. We then added 400 microliters of 50% ethanol and then transferred it into a GeneJet genomic DNA purification column, or spin column, that was then inserted into a collection tube. We placed the spin column with the sample into the centrifuge for 1 minute at speed of 600. We then discarded the collection tube fluids and added 500 microliters of wash 1 to the spin column and then placed it back into the centrifuge for 1 minute at 800. We then again discarded the fluid in the collection tube and added 500 microliters of wash 2 into the spin column and placed it back into the centrifuge for 3 more minutes at maximum speed. We then collected the sample from the centrifuge and put the spin column into the original extraction tube and added 100 microliters of elution buffer and then incubated for 2 minutes at room temp, then returned it to the centrifuge for 1 minute at speed 800. When that was complete, we discarded the spin purification column and stored the purified DNA that we extracted at negative 20 degrees Celsius. Next, we did a PCR, or a polymerase chain reaction, to amplify and target our region 18S SSU RNA gene. We used a PCR tube and made a mixture using 12.5 microliters of tag polymers, 10.5 microliters of H2O, 1 microliter of PF1 forward primer, and 1 microliter of FAD4 primer. Then lastly, we added 1 microliter of the DNA sample into that PCR tube. We then placed the PCR tube containing the sample and the mixture into the programmed thermal cycle. We set the lid to less than or equal to 95 degrees Celsius with an annealing temperature of 50 degrees Celsius. The thermal cycle will then cool to refrigerate the sample for a day and a half. The annealing temperature means heating and cooling double-stranded nucleic acid in order to separate strands and induce combination with complementary strands. We had to replicate this lab two more times to get the quality of purification that we desired. Next, we did gel electrophoresis to separate mixtures of DNA, RNA, and proteins according to their molecular size, and then to extract DNA bands from the gel. First, we weighed 0.6 grams of agarose and put it into a 225 microliter flask. We then measured 60 milliliters of VSB buffer into a cylinder. Next, we added that 60 milliliters of buffer into the 225 microliter flask containing the agarose and mixed the components. When the components were mixed properly, we microwaved the flask containing the solution for 1 minute, being sure to avoid boiling. When we got it out of the microwave, we added 1.2 microliters of CyberSafe to the solution to add dye to the gel. Next, we got the gel tray and placed the comb in the casting tray and poured 30 milliliters of gel solution into the casting tray and let the gel solidify for 30 minutes. Next, we filled that gel tank with buffer until it was completely covering the gel. We set the power source to 100 volts, running it negative to positive because DNA has a negative charge. We removed the comb from the casting tray, peeled off the backside pieces of the casting tray, and pipetted 10 microliters of the 5 individual PCR samples into the wells, leaving one well empty for 10 microliters of DNA ladder directly next to the samples. We placed the lid on the gel tank and turned the power source on to 100 volts for 20 minutes. After 20 minutes passed, we turned off the power source and carefully pulled the gel dams off the casting tray. Next, we slid the gel off the casting tray and onto a blue transilluminator. Using clean razors, we cut the successful DNA bands and placed them into 1.5 milliliter tubes to be put into the freezer at -20 degrees Celsius. This lab for gel electrophoresis was done 3 times so we could gather more samples of purified DNA to get a greater chance of gathering our protists. Next, we did a PCR cleanup of the gel extractions to purify the DNA segments in preparation for cloning. We began by getting an empty 1.5 milliliter tube and weighing it, and then weighing the 1.5 milliliter tube containing the gel extraction, and did a 1-to-1 mixture of binding buffer to the sample. We incubated this gel mixture at 50 to 60 degrees Celsius for 10 minutes until the gel was completely dissolved. Next, we transferred 800 microliters of the solubilized gel solution into a spin column, then placed it in the centrifuge for 1 minute at 600 speed. We removed the spin column after that 1 minute and added 700 microliters of wash buffer to the spin column, placed it back in the centrifuge for 1 minute at 1200 speed, and then emptied the follow-through tube. We discarded the follow-through again and placed the spin column back in for another round at 1 minute for 1200 speed again. Then we took the spin column back out, discarded the follow-through, and then placed the spin column into a 1.5 milliliter tube and added 50 microliters of elution buffer and placed it back into the centrifuge for 1 more minute at 800. We retrieved the tube with the sample and the elution buffer from the centrifuge and stored the purified DNA at -20 degrees Celsius. Next, we did bacterial selection and cloning. Prior to the beginning of this lab, we used a TA Topo cloning kit to clone our target gene. First, we retrieved the purified DNA sample, which we'll call HA1, and got another 1.5 milliliter tube, which we'll label H1C. Using the empty 1.5 milliliter tube, we pipetted 1 microliter of PCR4 TOPO vector, 1 microliter of salt solution, 4 microliters of the purified DNA extracted from the successful gel band. We allowed this mixture to incubate for 15 to 20 minutes at room temperature. After incubation, we pipetted 4 microliters of the sample into an E. coli competent cell tube, which we'll call H1E. Immediately incubate H1E in ice for 5 minutes, then place it into a heat shock dry bath with water at 42 degrees Celsius for 30 seconds. When we removed the sample from the heat shock, we added 125 microliters of SOC into the sample tube and returned it to ice for 30 more seconds. When the time was up, we removed the sample and placed it in the shaking incubator at 37 degrees Celsius for 1 hour. While the sample was incubating, we got a dish and labeled the bottom with the initials of the group. When we removed the sample from the shaking incubator, we pipetted sample from the H1E and spread it around the dish using a sterile spreader. We then let the plate containing the sample incubate at 37 degrees Celsius for 24 hours. Next, we did cloning and TOPO screening. Now we use a PCR reaction to extract and amplify our target gene from the E. coli colonies that we harvested in our plates. We began by retrieving the group plate and got 4 empty PCR tubes and labeled them individually. Using a pipette, we added a mixture to each PCR tube, which consisted of 10 microliters of TAG polymerase, 2 microliters of M13F primer, 2 microliters of M13R primer, and 6 microliters of H2O. Next, using the pipette, we picked 4 individual colonies from our dish and placed each sample into a different PCR tube. We then put those PCR tubes containing the colony samples into the PCR machine and ran the reaction to topo screen PCR setting for 2 to 3 hours. After this, we ran another gel electrophoresis to see if our cloning was successful. Then we ran another PCR on our best 3 colonies to purify and amplify our gene sequence for protists. We did a final PCR where we collected 3 PCR tubes and labeled each of them, and then one 1.5 milliliter tube and labeled that MMO for master mix. We added the master mix to the tube labeled MMO, which consisted of 40 microliters of TAG polymerase, 8 microliters of M13F primer, 8 microliters of M13R primer, and 24 microliters of H2O. We added 20 microliters of the master mix to each PCR tube. Next, we chose the 3 best colonies from our dish and put the individual samples into PCR tubes and let them sit in the master mix for 3 minutes. Then we placed the 3 PCR sample tubes into the PCR thermal cycle for 2 hours at 50 degrees Celsius for the annealing temperature to attempt to amplify the gene sequence further. When the PCR thermal cycle was complete, we did a final gel electrophoresis lab using the PCR samples and picked our most successful PCR gel bands and put them into PCR tubes to be given to the professor for further screening.